Welcome. My name is Dr. Hazrul Hisham bin Hashim. I am a clinical microbiologist from the Department of Medical Microbiology and Parasitology. This video is created as part of the GMT 205 course in the Year 2 MD program in University Science Malaysia. The title of this video is Laboratory Diagnosis of Infectious Disease. As a brief overview, when clinicians send samples to the microbiology lab to identify the microorganism causing infection in their patients, we can identify the microorganism through various methods such as through direct detection, culture, serology, and nucleic acid detection. For section 1 is specimen processing and identification. As we all know, the quality of the results depends very much on the quality of the specimen which the clinicians send to the lab. So, when the clinicians want to order a test, they should know what type of container or transport medium they should use to send the sample. For viral culture, they need to use viral transport medium. For blood culture, they need to use the backpack bottle. For serology, they need to use the plain bottle. For PCR, they need to send the sample in the EDTA bottle. And for transportation of swabs, they need to send the swabs in stored or AMIS transport medium. To send stool culture, they need to send in caribulary medium. And for stool culture for Vibrio cholera, they need to send the sample in alkaline peptone water. So, sometimes when the specimen is sent to the lab and it is rejected, there are various reasons why the specimens are rejected. For example, in the case of urine sample, it is because the urine is leaking. We cannot accept the sample because the sample may be contaminated. And then also the sample may be rejected in the case of blood culture because the volume of the blood in the blood bottle is not enough, and because we cannot accept this sample because the yield of the blood culture is very dependent on the volume, and so when the volume is not enough, we may give false negative results also. And then for the case of serology tests, the sample may be rejected because the sample has been hemolyzed. Because the hemolyzed sample will interfere with spectrophotometric result. And also in the case of swab sample, we may reject it because it's put in a sterile container. Because as we know, we need to put the swab in the AMIS or steward medium to prevent it from drying out. For section 2, there is bacterial identification, serology, viral isolation and detection, and molecular. Bacterial identification include microscopy, culture, and the various biochemical tests done on the culture, followed by immunological tests and molecular tests. So there are various microscopy staining methods such as gram stain for bacteria, gram-positive and gram-negative bacteria, Ziehl-Neelsen stain for Mycobacterium tuberculosis and acid fast bacilli, Indian ink for Cryptococcus neoformans, potassium hydroxide staining for dermatophytes, lactophenol cotton blue for fungus such as Aspergillus species, immunofluorescence staining for viruses such as influenza virus, and electron microscope for viruses such as rotavirus. And then there are culture methods for lab identification, whereby we use various culture medium to grow the organism. So first there is the basal medium, which is a very basic medium which will grow almost all non-fastidious organisms such as Pseudomonas aeruginosa. And then there is medium such as McConkey agar, which is both selective and differential. It is selective for gram-negative organisms, and it can differentiate between lactose fermenter and non-lactose fermenter. So this is used mostly for gut organisms, or known as Enterobacteriaceae. And then there are blood agar, which is an enriched medium, which can also grow a lot of organisms, such as Staphylococcus aureus. And then there is chocolate blood agar, which is an enriched medium, for example, used in Haemophilus influenza. So this is a fastidious organism. And then there is Thea-Martin agar, which is an enriched medium to grow fastidious organisms such as Neisseria gonorrhea. And then for antibiotic susceptibility testing, we can use Mueller-Hinton agar. So we can use Mueller-Hinton agar to test for antibiotic susceptibility testing on most of the organisms such as Pseudomonas aeruginosa. And to differentiate between the Enterobacteriaceae, we can use the oxycolate citrate agar, which is a selective and differential medium, just like McConkey. And for fungus, we can use Saborodestos agar, which is a selective medium for fungus. And for urine microorganisms such as Proteus mirabilis, we use cysteine lactose electrolyte deficient agar. We use this CLED agar so that we can prevent from swarming in Proteus mirabilis. And then for microbial tuberculosis, we can use Ogawa medium or Lowenstein Janssen medium. And for Vibrio cholera, we can use thiosulfate citrate bile agar, TCBS, so that we can identify Vibrio cholera, which is a selective medium for Vibrio cholera. So this is an example of the bacterial culture. On the left, you can see big colonies, mucoid and lactose fermenter. So from this from this bacterial culture on McConkey, we can identify it as Klebsiella pneumonia. But we need to further confirm with some biochemical tests. And on the right is a culture of Pseudomonas aeruginosa, where we can see big colonies with irregular age and green pigment. So we can confirm this by a few biochemical tests. So biochemical tests, there are various biochemical tests which can be done. First is catalase test, which is used to differentiate between staphylococci from streptococci. Staphylococci are catalase positive, whereas streptococci are catalase negative. So the bacteria produce catalase which break down hydrogen peroxide into oxygen which we can see as bubbles in the water. And then Staphylococcus aureus also produce the enzyme which converts the soluble fibrinogen in plasma into the insoluble fibrin. So, which this enzyme is not produced by coagulase negative staphylococci such as Staphylococcus epidermidis. And then another biochemical test is oxidase test. Oxidase test is used to identify bacteria that produce cytochrome C oxidase. All bacteria that oxidase positive are aerobic. For example is Pseudomonas aeruginosa and Vibrio cholera. Another biochemical test is in the indole test, which is used to distinguish Enterobacteriaceae based on ability to produce indole from tryptophan. So we can identify E. coli, which is indole positive, and Klebsiella pneumonia is indole negative. And then another biochemical test is urease test. So we can identify urease producers such as Proteus species and Morganella species. And some bacteria such as Klebsiella pneumoniae and Enterobacter species are weak urease producers. Then we can also use other biochemical tests such as carbohydrate fermentation and oxidation. So we can use the oxidation fermentation test, triple sugar ion test, methyl red test, and Vogt-Proskauer test. So in the triple sugar ion test, EGA, it is the initial step in identifying Enterobacteriaceae. It acts on carbohydrates and results in acidification in the butt of the tube, and also you can see whether there are gas formation and also whether there are iron salts which react with H2S to produce insoluble black precipitates in the TSI agar. So in the agar there are the two parts. First is the butt portion, which is the anaerobic portion, and then there is the slant portion, which is the aerobic portion. So for TSI, you can see carbohydrate fermentation, whether there is acid production, in where we can see the butt change color into yellow for glucose fermentation, and yellow slant to indicate lactose or sucrose fermentation. And then we can see gas formation, where we can see bubbles or cracking or splitting of the agar, upward displacement of the agar, or pulling of agar from the wall. And also we can see H2S production, where there is black color in the butt of the tube, which indicates ferrum sulfate precipitation. So in this slide we can see the interpretation. So in tube 3 we can see that there is acid slant, acid butt, gas formation, and no H2S. And in tube C there is alkaline slant, acid butt, no gas, and no H2S. So based on this finding, we can say that the organism is, for A, it is Pseudomonas aeruginosa, B is Klebsiella pneumonia, C is Salmonella species, and D is Shigella species. But we need to further confirm with other tests. So for E. coli, we can observe and record the biochemical reaction from the when we see the tube. So for E. coli is, we can see acid slant, acid butt, gas, and no H2S. And for Salmonella typhi, you can see alkaline slant, acid butt, no gas, and H2S present. So currently there are more modern tests with using more expensive machines which currently introduced into the lab. So in some hospitals, uh, they have access to these more modern machines, and so these modern machines they use extended panel of biochemical tests where they use a huge range of biochemical tests to identify the organism. So it is an automated machine to and used for microbial detection and also for antibiotic susceptibility testing, and it's very rapid. But the disadvantage is the high cost. So some hospitals may not be able to purchase such equipment and they just use the conventional tests. So when we have identified the organism, we need to do antimicrobial susceptibility testing to measure the susceptibility of an isolate to the antibiotic, and so that clinicians can prescribe targeted antimicrobial therapy. So there are various methods such as the conventional method, which is this diffusion Kirby-Bauer method, and the more recent Vitek 2, the automated machine. So for the conventional method, we use Mueller-Hinton agar or Mueller-Hinton blood agar antibiotic this, which the medium is lawn from the bacteria which we have isolated in the culture. So this is how the Mueller-Hinton agar is lawn, and then we place the antibiotic disc, and then we measure the area around the disc where to identify whether it is susceptible or resistant to the antibiotic. So a breakpoint is a chosen concentration of an antibiotic which defines whether a species of bacteria is susceptible or resistant to the antibiotic. After overnight incubation, the zone of inhibition is measured in millimeters and interpreted as susceptible, intermediate, or resistant using standard reference given by the CLSI, which is an international body which give guideline to for interpretation of this uh, antibiotic susceptibility testing test. So this guideline is updated almost yearly and provided to the lab by CLSI. So the interpretation can be susceptible, intermediate, or resistant. So susceptible means it is organism is inhibited by the recommended dose at the infection site by the antimicrobial agent. Intermediate means that the organism may require a higher dose of antibiotic for a longer period of time to be inhibited, and resistance means that the organism is not inhibited by the recommended dose at the infection site by the antimicrobial agent. So next we go to serology. So serology, we use the patient's serum to diagnose the pathogen causing the disease infection in the patient. So there are various serology methods, and for example is for leptospira we can use immunochromatographic test. For dengue also immunochromatographic test. For syphilis we can use the rapid plasma reagent, and we can use ELISA for a broad range of viruses, and Western blot is also used for HIV and hepatitis C as a confirmatory test. So for leptospira, we can use immunochromatography test. So we can detect the antibody towards the leptospira to diagnose leptospirosis. So all these tests, we need to make sure that before we can interpret whether it's positive or negative, we need to make sure that the sample is valid. So in the test kit, there is the first part is at the back is C, which means control. So if control is present, then it is valid. But if control is not present, then it's not valid. And such as in number E. And for D is not valid because uh, the test at 50 is not present, whereas in 200 the test is present, so it may be something wrong in the test, so it's invalid. So for A, we can see that the titer 50, the all A, B, C, all the control is present, and test for 50 and 200 is present, so we can say that the antibody titer is more than 200. For B, is more, the test for 50 is present, but for 200 not present, so it is between 50 to 100, the antibody titer. And for C, you can see the test line at 50 and 200 both are not present, so it is negative. So the red line must appear on each C line of the P and N in the detection window, and two red lines should appear on the test line P and N in the window. So the test line is written as T and the control line is written as C. So for intermediate, only positive red line of P window in the endemic region. It is necessary to do test again within 2 to 7 days. If in epidemic region, the result is interpreted as positive. And for negative, red line only appear on both control lines of the IgM IgG in the detection window, and no red line should appear on the test line. And for invalid, if red line appear only on N test or does not appear from at least one out of two C lines, the result is interpreted as invalid and we need to do the test again. Control line C is present. If the control line is not present, it's invalid. So NS1 is positive, and if NS1 is not present, it's negative, and if control line not present, it's invalid. So for dengue IgG IgM, control present, IgG present is IgG positive, control present, IgM present is IgM positive, and control IgM and IgG present is IgM IgG positive. So control present, no IgM, no IgG is negative, and no control, no IgG, no IgM is invalid. So the principle is that when the patient sample is put in the sample pad, so there is there is capillary action which pulls the sample through the test kit, so and you will bring the NS1 in the patient sample in the patient serum through the test kit, first to the immunogol and then to the anti-dengue virus NS1, and then to the goal IgM and IgG internal control. So when they reach to the place where there is anti-dengue virus NS1, or when it will bind and produce the red line. Next, we go to the test for syphilis, which is the rapid plasma reagent test. It refers to a type of test that looks for non-specific antibodies in the blood of the patient that may indicate that Treponema pallidum that causes syphilis is present. The term reagent means that this test does not look for antibodies against the actual bacterium, but rather for antibodies against substances released by cells when they are damaged by Treponema pallidum. So that is why this test is called as a non-treponemal test. So when we do this test, and we need to confirm by another test, which is a confirmatory test such as VDRL or TPHA. So the patient's serum is mixed with cardiolipin on a plastic coated card that can be examined with the naked eye. So when we see clumping, so it means that it is reactive, and then we can see the titer and record the titer. So for number 1 there is non-reactive, number 2 is weakly reactive, and number 3 is strongly reactive. We can see the clumping. So next serological test which is used in the lab is the enzyme-linked immunoassay ELISA, a 96 well plate based assay which is used for detecting and quantifying substances such as peptides, proteins, and antibodies. So there is antigen and antibody reaction. So they bind together, and an enzyme conjugated with the antibody reacts with the colorless substrate to generate a colored product. And specific substrates are used which are hydrolyzed by the above enzymes to give the colored end product, and this is measured by the machine so that we can interpret whether it is positive or negative. So there are 3 types of ELISA: the direct assay, indirect assay, and the sandwich. So in the direct assay, the antigen is bound to the microtiter plate, and then we detect the antibody in the patient's serum. So the enzyme cleaves the substrate and produce the color. So in the indirect assay, the antigen is bound to the primary antibody, and we detect the antibody, and the secondary antibody binds to the primary antibody, and the enzyme cleaves the substrate to produce the color. And then for sandwich assay, the captured antibody is bound to the microtiter plate, so we detect the patient antigen which attached to the secondary antibody, and the enzyme cleave the substrate to produce the color. So for HIV, after we have done the ELISA, we can confirm with the Western blot test or Inolia test. For Western blot test, we can use for detection of HIV viral proteins. So HIV proteins are separated by PAGE, polyacrylamide gel electrophoresis, and blotted onto nitrocellulose paper strip. So the test serum is allowed to react with the strip. Antibody in the serum combines with HIV protein fragments. Position of the color band on the strip indicates the antigen which antibody had reacted. So positive results should show at least 2 of the 3 following bands: P24, GP41, and GP120 or GP160. So this is the HIV virus. There are a lot of proteins. So P18 is the core matrix, P24 is the core shell or capsid, P31 is endonuclease, P40 is core, GP41 is transmembrane glycoprotein, P55 is core precursor or P51 or reverse transcriptase, P65 is reverse transcriptase, GP120 is outer envelope or surface glycoprotein, and GP160 is the envelope precursor. So all these are components of the virus. So we can detect these proteins of the virus using Western blot, which is present as bands on the test strip. So we can see what bands which are present, and based on the bands we can say that what proteins are present, and if 2 or 3 are present based on the criteria given by the test kit provider on WHO guideline, we can diagnose it as HIV positive or negative. So the next method is uh, viral isolation and detection. So we can use viral culture, viral microscopy, and detection. So for viral culture, we can use it to grow the virus in cell cultures, and from this, when the virus grow in the cell cultures, we can observe cytopathic effect, immunofluorescence microscopy, and we can do plaque formation, we can do hemagglutination assay, neutralization tests, and metabolic inhibition tests. To all these tests we can do to identify the virus. So there are various viruses which can grow in the cell culture, such as herpes simplex virus, adenovirus, RSV, parainfluenza virus, mumps, Japanese encephalitis, and dengue virus. But there are also some viruses which cannot be cultured, such as rubella, varicella zoster, or Coxsackie virus. So this virus cannot be cultured. So we need to use other tests, such as serology, to diagnose. Between 7.2 to 7.4 in a humid environment, and temperature between 35 to 37. Lower temperature, 33 degrees for respiratory viruses. So this is the environment for the viral culture. So when we culture the virus in the cell culture, we observe the cytopathic effect, which is the visible results of viral infection in the cell culture. So we can see cytopathic effect of infected cells using the inverted light microscope. So we can see rounding or detachment from the plastic fuzz. We can see syncytia, where 3 or 4 cells fuse together and become one giant cell with multinucleated giant cell. So this is seen in herpes simplex virus, syncytial virus. We can see syncytia, and then we can see shrinkage in enterovirus. We can see increased refractility. We can see aggregation in adenovirus. We can see loss of adherence in certain oncogenic viruses. We can see cell lysis. Also, we can see inclusion bodies in the cell culture. So some develop intracytoplasmic inclusion bodies, such as in rabies, the CPE is eosinophilic with Negri bodies, and for adenovirus, it is intracytoplasmic inclusion body with basophilic. And for intranuclear inclusion bodies such as herpes simplex virus or varicella virus, we can see it is eosinophilic intranuclear inclusion bodies. And for adenovirus or CMV, we can see basophilic intranuclear inclusion bodies. So for CMV, we can see the characteristic owl eye appearance. So other tests such as hemadsorption tests can also be done for influenza virus. So this is a cytopathic effect where in the first on top left box we can see the uninfected cell culture, and in the bottom right is what we see after 24 hours. So this is what is we observe, and this is called cytopathic effect, which is a virus-induced cell damage. So this cytopathic effect is different between the different viruses. So the type of CPE will indicate what type of virus uh, is uh, is present. So another method is immunofluorescence test. So for virology, we can use immunofluorescence test where we use specific antibodies from the test kit which bind to specific viral antigens in the infected cells. So the bound antibodies are stained with fluorescence dye and subsequently visualized as green spots under a fluorescence microscope. So the next test is latex agglutination test, which we use for stool samples such as rotavirus or adenovirus. So a specific antibody coat the surface of the latex particle, and when stool sample containing the virus specific antigen is mixed with the milky appearing sensitized latex, it causes visible agglutination. And then the next method which can be used is electron microscope, which is capable of demonstrating the virus directly. So the advantage is we can visualize new viruses or pathogens, and can visualize viruses which cannot be cultured, such as hepatitis. So we in Malaysia we discovered the Nipah virus through electron microscope. So it was a new virus at that time, and people were thinking that the outbreak was caused by Japanese encephalitis at the time, but it was subsequently proven to be not Japanese encephalitis, but a new virus, which is called the Nipah virus. After doing electron microscope, whereby on microscope we can see that it is a paramyxovirus and not the Japanese encephalitis virus. So the disadvantage is electron microscope is not sensitive. It requires high virus concentration, more than 10 to the power 6 of particles per mil, and it requires expensive equipment and trained technician. So it is very rarely done. But when there is an outbreak of new viruses, then it's very useful. So specimens include feces such as rotavirus, adenovirus, astrovirus, vesicle fluids, and skin scraping. So the sensitivity and specificity of electron microscopy may be enhanced by immune electron microscopy. The sample is treated with specific antisera before being put for electron microscopy. Viral particles present will be agglutinated and thus congregate together by the antibody. So this is electron microscope of rotavirus, where we can see the characteristic wheel structure. So from this wheel structure on electron microscopy, the virus got its name rotavirus, where rota in Greek means wheel. And this on the right is the electron microscope electron micrograph of influenza virus. The structure of the virus can either be filamentous or spherical. So in this electron micrograph, it is a spherical. And then on the left is adenovirus. So we can see spikes on the capsid which aid attachment on target cells. And on the right is hepatitis B virus, which exists in different forms, including spherical and filamentous forms. So next section is molecular diagnostics. So there are a lot of molecular tests which can be done. Among them is PCR, whereby we use nucleic acid amplification. So the principle of molecular diagnostic is each microorganism contains specific DNA or RNA gene sequences that are unique to that organism. By detecting these unique gene sequences, we can identify the microorganism. Molecular diagnostic is based on the detection of specific gene sequence contained in microorganisms. The presence of an organism's gene sequence in a patient sample allows for the positive identification of the organism in the patient. Molecular diagnostic is detection of specific gene sequence which are detected after amplification through amplification assay, PCR. PCR techniques enzymatically multiply a specific gene sequence exponentially, resulting in the production of billion copies of the sequence in a short period of time. The amplification product or amplicon is detected by agarose gel electrophoresis to definitively identify the organism present in the sample. So the steps is the DNA extraction from the sample, and then we run the PCR, and after that we run the PCR product on agarose gel, and then we visualize the PCR product under UV light. So this is the step. First we run the sample in the machine, then we do the agarose gel preparation, and we load the sample, and we do electrophoresis, and then we visualize the PCR, and we can see the bands on gel electrophoresis. So on gel electrophoresis, we can see the PCR product, and which is stained with ethidium bromide. So on the right, we can see the ladder, and in the middle, we can see the PCR product. So we can compare with the PCR product with the ladder, and which base pairs number which is present to identify the organism. So now we go to the last section, which is section 3 of pathogens. So there are various pathogens which causes infection in our patients in the hospital. So in this section, we will discuss some of the more common organisms. How in the lab we use all the various tests which we have learned just now to identify the organism. So for Staphylococcus aureus, we first do microscopy. We see there is a gram-positive cocci which grows well aerobically and in CO2 in the rich atmosphere at a temperature range of 35 to 37 degrees. On blood agar, we can see it as a creamy colonies 1 to 2 millimeters in diameter. In microscopy, we can see that it is a gram-positive cocci arranged in clusters. So the disease caused by Staphylococcus aureus is it causes purulent skin infections such as boils, abscesses, impetigo, and scalded skin syndrome. Systemic infections such as pneumonia, osteomyelitis, or endocarditis. Hospital-acquired infection of surgical wounds or central lines. Food poisoning by releasing toxins into blood. Toxic shock syndrome by releasing toxins into the blood. So to treat this infection, we can give cloxacillin, erythromycin, and for the resistant Staphylococcus aureus, or known as MRSA, we can give vancomycin. Next, we go to streptococci. Streptococci are classified by their hemolytic action on blood agar through Lancefield grouping. So, the hemolytic reaction can be beta hemolysis, where we see complete clearing of blood agar, alpha hemolysis, where there is incomplete hemolysis, causing a greenish discoloration of the agar, and gamma or non-hemolysis in the blood agar. After that, we can do Lancefield grouping to group the streptococci into group A, B, C, D, F, and G. Some streptococci do not have Lancefield grouping. So this is the types of hemolysis. There is beta hemolysis, where we can see clear zones around the colonies in blood agar. There is alpha hemolysis, where there is green zone around the colonies. And there is also gamma or non-hemolysis, where there is no change of blood agar around the. So for Streptococcus group A, or known as Streptococcus pyogenes, on gram stain, you can see that there is gram-positive cocci in chain. And on culture, on blood agar, we can see that it produces a white to gray colony 1 to 2 millimeters in diameter, surrounded by zones of complete hemolysis or beta hemolysis. In some strains, it may become also seen as mucoid in the, strains which produce capsule. So the diseases caused by Streptococcus group A, it is the most common infection in streptococcal pharyngitis. It causes superficial skin and soft tissue infection, including impetigo, erysipelas, and cellulitis. It causes severe life-threatening infections, including scarlet fever, bacteremia, pneumonia, necrotizing fasciitis, myonecrosis, and streptococcal toxic shock syndrome. It can also cause post-infectious sequelae, including acute rheumatic fever and post-streptococcal glomerulonephritis. So the antibiotic of choice for Streptococcus group A infection is penicillin. Or you can also use topical antibiotics such as bacitracin and mupirocin in the case of impetigo. Next, we go to Streptococcus group B, also known as Streptococcus agalactiae. So on microscopy, the gram staining, we can see gram-positive cocci in chain. And on blood culture, blood agar, the colony is 3 to 4 millimeters in diameter, grayish-white, the colonies are flat and mucoid and surrounded by a narrow zone of beta hemolysis. Streptococcus group B is normal flora in the genital tract of pregnant women, but it can cause early onset neonatal sepsis, invasive hematogenous infection in compromised hosts. So the antibiotic of choice is penicillin. Next is viridans group of streptococci, such as Streptococcus mitis. So there are a few species of viridans group, Example is Streptococcus mitis, Streptococcus mutans, and others. So on gram staining, we can see gram-positive cocci in chain, and on blood agar, we can see alpha hemolysis surrounding the white to gray colonies. So the species under this group is also include Streptococcus aeruginosus, Streptococcus mitis, Streptococcus mutans, Streptococcus salivarius, and Streptococcus sanguinis. So for identification, on blood agar, we can see alpha hemolysis in Streptococcus pneumoniae and also in this virulence group of Streptococci. So that's why we need to use the antibiotic optocin this when we culture the sample on blood agar so that we can differentiate from Streptococcus pneumonia. So the virulence group of Streptococci is usually a normal flora from mucosal surfaces, particularly the oral cavity. So it rarely causes disease, but in certain patients who have infective endocarditis, then we need to treat. And the antibiotic of choice in that situation is penicillin. Streptococcus pneumoniae, on gram staining, we can see gram-positive lancet-shaped Diplococcus. And on blood agar, we can see mucoid with glistening colonies up to 5 millimeters with alpha hemolysis. So the characteristic of the colonies on blood agar, we can see depression in the center, also known as target shape. So on blood agar, we can identify it. It is alpha hemolysis and it is optocin sensitive. So the diseases caused by Streptococcus pneumoniae include pneumonia and bacterial meningitis in children younger than 5 years and older adults worldwide. So the antibiotic of choice is penicillin, and an alternative treatment for penicillin-resistant Streptococcus pneumoniae are streptozan or cefotaxime. So on this slide, we can see virulence group of Streptococci which is resistant to optocin, and on the right we can see Streptococcus pneumonia which is sensitive to optocin. We can see there is clearing around the optocin disc. Next we go to Enterococcus species. So on gram staining, we can see there is a gram-positive cocci in pairs or short chain. The blood agar, it is 1 to 2 millimeters in diameter, and gamma lysis or non-hemolysis. So the species mainly is Enterococcus faecalis and Enterococcus faecium. And the disease it causes include urinary tract infection, wound infection, especially abdominal wounds, endocarditis, and meningitis. The antibiotic of choice is ampicillin and penicillin. Next, we go to the Enterobacteriae. First one is E. coli, Escherichia coli. So on microscopy, we can see that it is a gram-negative bacilli. And on McConkey agar, we can see smooth pink colonies, and the color of the medium change because it is a lactose fermenter. So on microbiology, it is aerobic and facultative anaerobe. Gram stain is gram-negative bacilli, and on McConkey agar, it is a smooth pink colonies, lactose fermenter. Disease causes include UTI, example cystitis, It also causes wound infections, peritonitis, sepsis, and meningitis in neonates. And it also causes the diarrhea diseases. E. coli strains associated with diarrheal diseases include ETEC, EPEC, EIEC, and EHEC. So these strains of E. coli, they produce toxins which cause disease to the patient. And antibiotic therapy include cefuroxime, amoxicillin clavulanic acid, Bactrim, and piperacillin tazobactam. Next, we go to Klebsiella pneumoniae. It is an aerobe and facultative anaerobe. The gram stain, we can see gram-negative bacilli. On monkey agar, we can see large gray white mucoid colonies, which are lactose fermenter. Change the medium into pink. And the disease it causes include chest infections, bronchopneumonia, and lung abscess. It also causes hospital-acquired infections of the wound and of the urinary tract. So antibiotic therapy can be given include cefuroxime, amoxicillin clavulanic acid, Bactrim, and piperacillin tazobactam. Next, we go to Pseudomonas aeruginosa. It is a non-sporing, motile, oxidase positive. On gram staining, we can see gram-negative bacilli. On McConkey, we can see pale colonies. And on neutrine agar, you can see green pigmented colonies. The disease it causes include skin infections, especially in burn patients, patients with ulcers, and pressure sores. It can also cause urinary tract infections following catheterization. It can cause respiratory infections in immunocompromised patients and patients with cystic fibrosis, and it can also cause external ear infections. The antibiotic of choice is ceftazidime, piperacillin tazobactam, and cefepime. Next, we go to Haemophilus influenzae. On microscopy, on gram staining, we can see small pleomorphic gram-negative rods or coccobacilli with random arrangements. On chocolate agar, you can see the colonies are flat, grayish-brown with diameters of 1 until 2 micrometers. We need to culture the organism on chocolate agar because it is a fastidious organism. Clinical significance, it is an obligate human pathogen, most often causes infections in the respiratory tract, occasionally causes bacteremia and meningitis. Haemophilus influenzae type B was formerly known as the leading cause of bacterial meningitis in children. The antibiotic of choice is ceftriaxone, especially for meningitis, and cefotaxime, also for meningitis. You can also use ceftazidime, ampicillin sulbactam, fluoroquinolones, or azithromycin. Next, we go to Neisseria gonorrhea. On gram stain of urethral discharge, we can see it is a gram-negative diplococci intracellular. So we can differentiate with Neisseria meningitidis, which is not intracellular. We can see Neisseria meningitidis outside of the cell. But for gonorrhea, it is a obligate intracellular. On gram staining from the culture, gram-negative singles or in pairs, this arrangement is produced because the cells divide in the same plane and the pairs separate as they divide. And on Theomartin agar, the colonies vary in size from 1 to 4 millimeters after 48 hours. The colonies are smooth and non-pigmented. Again, it is cultured on chocolate agar, on Thayer Martin agar because the organism is fastidious. The clinical significance of Neisseria gonorrhoeae. This is a sexually transmitted bacteria. It can cause pharyngitis, proctitis, urethritis, prostatitis, orchitis, pelvic inflammatory disease, conjunctivitis. Disseminated Neisseria gonorrhoeae infections can occur, resulting in endocarditis, meningitis, or gonococcal dermatitis arthritis syndrome. Also, it can cause dermatitis arthritis syndrome present with arthralgia, tenosynovitis, and painless non-pruritic dermatitis. The antibiotic of choice is ceftriaxone plus azithromycin or doxycycline if chlamydia cannot be ruled out. You can also use cefixime plus azithromycin or doxycycline if chlamydia not ruled out, or we can also use fluoroquinolones such as ciprofloxacin. But it's not recommended as empiric treatment for gonococcal infections in many parts of the world because of resistance. Alternative antibiotics include spectinomycin or cefotaxime. Next, we go to Proteus species. On gram staining, we can see gram-negative bacilli with no arrangement. On culture, we can use blood agar. We will see swarming. And on McConkey agar, we can see non-lactose fermented colonies. And the clinical significance, there are 3 species from the genus Proteus are opportunistic human pathogens: Proteus vulgaris, Proteus mirabilis, and Proteus panneri. The main infections caused by these organisms are urinary tract infections and wound infections. The urease enzyme of Proteus and others are able to cause alkalinization of the urine and increase risk of renal stones. The antibiotic of choice for uncomplicated UTI in women can treat with oral antibiotics. Other antibiotic of choice is quinolones, Bactrim, and for complicated UTI, we need to use parenteral antibiotics such as ceftriaxone, quinolone, gentamicin plus ampicillin, or aztreonam. For Salmonella typhi and Salmonella species, it is a facultative anaerobe which is motile. On gram stain, we can see gram-negative bacilli, and on McConkey or the oxycholic citrate agar, we can see small, circular, colorless, non-lactose fermented colonies. And some strains show black center due to H2S formation on DCA agar. The disease it causes include acute gastroenteritis resulting from a foodborne infection by non-typhoidal salmonella such as Salmonella typhimurium and Salmonella enteritidis. And we can see enteric fever resulting from bacterial invasion of the bloodstream by typhoid by Salmonella typhi and paratyphoid by Salmonella paratyphi A, B, or C. Antibiotic therapy for Salmonella species, we can give ampicillin, ciprofloxacin, ceftriaxone, usually self-limiting and does not require antibiotics. For Salmonella typhi, we can treat with ciprofloxacin or ceftriaxone. For Shigella species, it is a facultative anaerobe, non-motile. On microscopy, gram stain, we can see a gram-negative bacilli. On McConkey or DCA agar, you can see small to medium-sized, colorless, non-lactose fermented colonies. The diseases caused include shigellosis, bacilli dysentery, acute gastroenteritis resulting from a foodborne infection. Antibiotic therapy include ampicillin, amoxicillin clavulanic acid, ciprofloxacin, and cefuroxime. Next, we go to Vibrio cholera. It is a facultative anaerobe, highly motile. We can see rapid darting movement by hanging drop method. On gram stain, we can see gram-negative bacilli, curved shape. And on TCBS agar, we can see small to medium-sized golden yellow colonies for Vibrio cholera and green colonies for Vibrio parahemolyticus. So it causes cholera, and the antibiotic of choice is doxycycline. Next, we go to Campylobacter jejuni. It is a microaerophilic organism. On gram staining, we can see gram-negative bacilli, pleomorphic, comma, or S-shape. And on Campi agar or CCDA agar, incubated in microaerophilic conditions at 42 degrees Celsius, we can see small size mucoid grayish flat colonies. So it causes AGE, acute gastroenteritis, and for antibiotic therapy, usually is self-limiting and does not require antibiotic. Mycobacterium tuberculosis, we can use Ziehl-Neelsen staining because it is a acid-fast bacilli and cultured on Ogawa or lower St. Johnston medium, where we can observe it as a dry, creamy, buff colonies. So the disease, Mycobacterium tuberculosis, is the causative agent for tuberculosis. It is the most common form of infection, is pulmonary tuberculosis. 90% of all the TB cases, and the other 10% present as extrapulmonary TB, which include TB lymphadenitis, TB meningitis, musculoskeletal TB, TB gastrointestinal infection, and others. So the antibiotic of choice for we usually give a combination of isoniazid, rifampicin, pyrazinamide, ethambutol, and streptomycin in phases. First in the intensive phase, and then in maintenance phase, according to the WHO protocol. Next, after finishing about the most common bacteria, now we go to fungi. So fungi can be divided into molds and yeast. So molds can be aseptate and septate. So the aseptate molds have aseptate hyphae, and they can cause subcutaneous mycosis such as, and the organism examples include Basidio-Bolus species, Conidio-Bolus species, Rhinosporidium seaberi, and also systemic infections such as Rhizopus species, Absidia species, and Mucor species. And then there are molds which produce septate hyphae, so they can be monomorphic or dimorphic. The monomorphic include Glycosporium baegelii, dermatophytes, Malassezia, furfur, and Pedrea hortae. Others include Cladosporium, Phyalophora, Madurella, and Acrimonium. Dimorphic molds include the systemic causes include histoplasma, blastomyces, and paracoccidiosis, and superficial infection include Malassezia furfur. And then yeast can be divided into without pseudohyphae and with pseudohyphae. Without pseudohyphae include Cryptococcus neoformans, which causes a systemic infection. And there are yeast which causes cutaneous and systemic, which include the various types of Candida species. So for Candida, the most common is Candida albicans. On gram staining, we can see gram-positive, ovoid-shaped cells, and some of the cells have budding. On Seborrhoeides agar, we can see cream-colored, smooth surface, waxy. So the common species of Candida include Candida albicans, Candida glabrata, and others. Diseases include cutaneous and mucosal candidiasis, chronic mucocutaneous candidiasis, opportunistic infection, which is infections involving all organs, and also disseminated candidiasis. Treatment of choice is fluconazole, amphotericin B, and echinocandins such as esofungin. For Cryptococcus neoformans, on India's in staining, we can see that there is a capsule present as a halo around the uh, Cryptococcus due to the capsule. And then on Seborrhoeides agar, we can see dark, cream-colored, smooth, moist, shining mucoid colonies. So the Cryptococcus pneumoniformans, it causes meningitis, cryptococcal meningitis, pneumonia, pulmonary cryptococcosis. So the treatment include amphotericin B and flucytosine. The type of mold by observing the structures. On culture Seborrhoeides agar, you can see that Aspergillus flavus produces colonies which are granular, flat, often with radial grooves, yellow at first but quickly becoming bright to dark yellow-greenish with age. And microscopy, we can see on lactophenol cotton blue staining, we can see conidial heads which are typically radiate, later splitting to form loose columns, mostly 300 to 400 microns in diameter, biseriate but having some heads with highlights borne directly on the vesicles, uniseriate. Common species of Aspergillus include Aspergillus flavus, Aspergillus fumigatus, and Aspergillus niger. Diseases include allergy, pulmonary aspergilloma, invasive aspergillosis. The treatment of choice is amphotericin B and voriconazole. That's all for my video today. I hope you enjoy microbiology. And if you have any question, please contact me at any time. Thank you.